Hello everyone, welcome to the new session of Agriculture Microbiology, and in this session we are studying about the bacteria, right? We have already covered some part of the bacteria, right? In this second part we are going to cover up the cellular component of the bacterial cell. So in this we will study about cytoplasm, different cellular organelle, though there are not much organelle present in the bacterial cell, but yes, mesosomes are there, ribosomes are there, nucleoid and plasmids. So all these structure that we are going to study. Also the flagella, locomotories, flagella, cilia and pili structure, right? So all this thing we are going to cover up as a cellular component of the bacterial cell. Now other part, that is the reproduction in bacteria. So there are two different types of bacteria, that is the asexual reproduction and the sexual reproduction that takes place in the bacteria. So that we are going to cover up here. And also the common bacterial diseases that occur in the plants. So all this we are going to cover up in this session. So the cytoplasm. It is a crystallocolloid complex that forms the protoplasm excluding its nucleoid. So what is nucleoid? First of all, let us understand that nucleoid is suppose this is the bacterial cell. In the center, a single circular chromosome is there. Single circular chromosome is there. So that is called as nucleoid, and it is not covered, or you can say the bound by the membrane like structure as we can see in the case of eukaryotic cell. So that is called as nucleus, that is true nucleus. But here this nucleoid, it is not a true nucleus because it is not covering this single circular chromosome from the outside. So it is present in the cytoplasm itself, right? So what is cytoplasm then? Excluding this nucleoid, whatever this crystallocolloid substance, liquid substance that is present in the cell, that is called as a cytoplasm. Right? And the membrane bound cell organelles are absent. No cell membrane bound organelle is present in the cytoplasm. So this is one of the very important part. Cytoplasm streaming is absent. What is cytoplasm streaming? So if we see the structure of amoeba, that is a eukaryotic cell, it is having the nucleus. Cytoplasm is moving like this. So that is called as cytoplasmic streaming. That is not found in bacterial cell. So that is totally absent. So this characteristic is absent in the case of bacterial cell, that is cytoplasmic streaming. These points are very important because sometimes the multiple statement question comes, which of the following statement is false or true about the bacterial cell. So that in that case you must remember this, that cytoplasm streaming is totally absent in the case of prokaryotic or the bacterial cell. Right? Various structures are present in the cytoplasm and are as follows. We are not having any cellular membrane bound organelles in the case of bacteria, but yes, there are various structures that are present. So let us see what are those. The first one is cell membrane of bacteria develop and in growth. So at some places the membrane, as you all know, the outermost is the slime layer, that is a type of glycocalyx, right? Then comes the cell wall. Here we can see this cell wall structure, right? And then comes the inner, that is the plasma membrane. So at some places this plasma membrane invaginate and form such type of structure like this. Now this structure is called as mesosomes, right? So this is the specialization of the cell membrane. It is you can say the cell membrane at some places get specialized and invaginate itself, and it contain respiratory enzyme. Very, very important. It is containing the respiratory enzyme and hence believed to be equal to the mitochondrion of eukaryotes, because what we see in the case of mitochondria, suppose this is a mitochondria. I'm drawing a rough figure and you can see the inner membrane of mitochondria invaginate itself and form a cristae-like structure like this. The cristae invagination is different and different types of bacteria, right? Sometimes it's straight, sometimes it is branched. So here I'm just drawing the simple one that is showing the cristae structure. This is the outer membrane of bacteria. This is just to understand what is the similarity between this mesosome and the normal bacteria that is present in the eukaryotic cell. So this is the inner membrane, right? And this inner membrane, it gives a ingrowth like structure, a finger like ingrowth like structure that is called as cristae, right? So here what you see, this inner membrane, it is forming an ingrowth, or you can say the invagination. So what you see here in the case of mesosome also, it is also having such type of invagination, right? And it is having some tubules or some vesicles like a structure, but this is came out from this plasma membrane also, in growth of the plasma membrane only, and this is called as mesosome. Now this is having a lot of you can say enzyme attached with it. Okay, so such type of enzymes are also present at the mesosome only. So this is the similarity between the mitochondria. Okay, so that is why it is called as a very, very important structure that is present in the prokaryote, and it resembles the mitochondria. That is why. Okay, so do remember this mesosome. This is the invagination of the cell membrane of the bacteria, right? It develops in growth in the cytoplasm itself, and it contains all those respiratory enzymes that is present at the mitochondria, that is present inside the mitochondria. Next is the ribosomes. So ribosomes are the small membranes, less ribonucleoprotein entities. Why it is called ribonucleoprotein entities? Because it is having two parts, rRNA, you can say simple RNA, and you can say protein. Two parts are there, and that is why it is named ribonucleoprotein. Protein is there, and ribonucleus structure, that is RNA, nucleic structure, right? Now if you see the type of ribosome that is here, it is having two subunit, smaller subunit and the larger subunit. This smaller subunit is of 30S type, and the larger one is that is of the 70S type, and the total this is the 50S, and it is called as 70S type. Now this smaller one, that is containing 16S RNA, and this one is having larger one, that is having 23S RNA and 5S RNA. 5 sRNA, 2 component of RNA is here. And that this is the peculiarity of this bacteria. And if you talk about the eukaryotes, then it is again, it is of 40S and 60S type. This is the smaller subunit and this is the larger subunit. Okay, so here the component is very different. That is a smaller one that is having 18S. Now you can compare it. It is having the 16S. Here it is having 18S, right? And in this also, what we see? We see that in the 60S type, we are having 28S RNA, 28S RNA and 5S RNA, 5S RNA and 5.8S RNA. So 3 RNA, 5S RNA, 5.8S RNA and 28S RNA. Now if you see the largest subunit of the prokaryotic ribosome, that is of the 50S type, it is having 5S RNA. So this 5S is present in both the type. Other things are not common. In 18S, that is present in the larger subunit in the eukaryotic cell. 16S is present in the smaller subunit in the prokaryotic cell. Now if we see the larger subunit, it is having of eukaryotic, it is having 3 different types, that is 5S, 5.8S and 28S. Now larger subunit of the prokaryotic, it is having 5S and 23S, right? And this 5S is only common in the 2 types of ribosome, that is 70S, 80S type, 80S type. So here the ribosome are of 70S in nature. I've just give you a rough figure that sometime this subunit can also be asked, right? What is the subunits of 70S type in the prokaryotic? It is 30S and 50S. Do remember this. Okay, so they are of 2 types of free ribosome, free in cytoplasm. So if we see the bacterial cell, ribosomes are free, that is present in the cytoplasm, that is called as free ribosome, and fixed ribosome attached to the plasma membrane. So here they are having the plasma membrane. At this plasma membrane, lots of this ribosome get attached. Okay, so they are called as fixed ribosome. So ribosomes in bacterial cell are of 2 types, free ribosomes and fixed ribosomes, right? So this is very, very important. Chromatophores, they are present in the photoautotrophic bacteria. Photoautotrophic bacteria means the bacteria that can do the process of photosynthesis. Okay, so in purple bacteria, they are covered by the normal membrane, while in the chromatophores, they are covered by the non-unit membrane. If it is a unit membrane, always in the bacterial cell, something which is bound by the membrane, it is a non-unit membrane. So you cannot say that this is a membrane bound structure. Very important. Right, so this chromatophores, they are covered by a non-unit membrane, means it is non-lipid. The membrane, it is not made up of lipid, protein membrane, right? So it is a membrane simply, but it is not a unit membrane. So that is not considered as a membrane bound organelle. So these chromatophores are present in that form also, and sometime they are present in the photoautotrophic bacteria. In purple bacteria, they are covered by the normal membrane, while in the chromatophores, they are covered by the non-unit membrane. So it represents the genetic material of the prokaryotes. Nucleoid, very, very important. Nucleoid, genetic material of the prokaryote. Why it is called as the genetic material? Because it contains single circular chromosome. This single circular chromosome circular chromosome that is made up of two things, that is DNA, okay, and here protein structure is not there, means histone is not there, no histone, histones is not present. Protamines are there, so its composition, its structure is quite different. A nucleosome type of structures is not found here because this histone is not present here, okay. So this is the one of the difference between the eukaryotic and prokaryotic DNA, circular DNA structure, right? It consists of a single circular strands of DNA duplex which supercoiled with the help of RNA and polyamines. So this is the polyamines. This is very important. Instead of histone, they are having the polyamines and RNA structure. So this chromosome structure, single circular chromosome structure, it is made up of DNA duplex, means double helical structure, as we can see here like this. Double helical structure is there, RNA is there, and polyamines is there. DNA prokaryote is considered as naked. Why it is considered as naked? There are two reasons. The number one reason, no membrane bound structure. Membrane bound structure. That is the nuclear membrane. Nuclear membrane that is totally absent. Nuclear membrane is totally absent. And the second thing, histone structure is not there. So histone gives it a very high level of coiling structure, and it invaginates this DNA in a very small place, a long stretch of DNA that can be present at a very small place. So instead of this, they are having a polyamine. So this histone is not there, right? So due to the absence of this nuclear membrane and histone, this is considered, this DNA of the prokaryote is considered as a naked. Other name is given to the nucleotide are the genophore prochromosome. Sometimes this name is also given to this nucleoid structure, so do not get confused with that. It means the same thing, that nucleoid, right? So genophore, prochromosome, nucleoid, it is all give the same. And this nucleoid means the primitive type of nucleus. What does this mean? It means the primitive type of nucleus. So the organism that first forms in the sea, they do not have the exact nucleus like structure which is present in the eukaryote. They are having the primitive type of nucleus that is called as nucleoid. Plasmids. So self-replicating extra chromosomal segment of double-stranded circular naked DNA. So if you see the bacterial cell, what you see in the bacterial cells, in the center you can find out the single circular DNA that is called as nucleoid, right? Nucleoid. Now there are some circular DNA like this, small circular, and they can replicate itself, self-replicating, right? Self-replicating extra chromosomal segment means this is the chromosomal segment. Now a part of this chromosomal segment, they are having small circular DNA structure like this, right? And they are self-replicating. They do not require this single circular DNA. That is the main chromosomal DNA of the this bacterial cell. That is the nucleoid. It do not require anything from that, right? So extra chromosomal segment, that is the double-stranded, circular, and naked DNA. Again, it is not covered by any membrane-like structure. So these are called as plasmids. These are called as plasmids. Independent of the main nucleoid, so it never get recombined with this, so it is independent of the main nucleoid. They may contain important gene for the resistance, fertility factor, and the NIF genes. So NIF genes, sometimes the bacteria that is involved in the process of nitrogen fixation. Okay, so they contain NIF genes. NIF genes means they are responsible for the nitrogen fixation. Nitrogen fixation. So the gene that is responsible for the nitrogen fixation is present where? It is present in this plasmid structure. It is present in the plasmid structure. And so, and this in the field of this agriculture, it is very, very important, because what they have done, and if we talk about the small bit of use of this plasmid, then biotechnologists have taken out this NIF genes from this plasmid of this bacteria, and they had incorporated into the main DNA of some of the plants that are commercially very important for the agriculture. Now that plant can do the process of nitrogen fixation with itself, right? So this is very, very important. Now there are certain genes that are responsible for the fertility. F gene. Fertility means sometimes pili structures are there, and this pili structure forming a bridge between the two cells, and the DNA is passes from this bridge, from this tube-like structure, from one to another bacterial cell. So this happened due to the formation of this sex pili, and this sex pili formation is encoded by where? F gene, that is also present on one of the plasmid. Okay, so that is why they contain the fertility factor. This is called as the fertility factor and the NIF genes. A part of this, they also contain gene for the resistance. Some of the plasmid, they contain tetracycline resistance, streptococcus resistance gene, right? So all these bacterial resistance genes are also, you can say, antibiotic resistant genes are present at the plasmid. So that is very, very important for the biotechnology respect. Biotechnology. Because these plasmids are used for making vectors. Now these vectors are the vehicles that can incorporate the desired gene from one organism to the another. So that we will study later. Okay, but here you have to remember the plasmid, that is extra chromosomal segment of the double-stranded circular naked DNA. Okay, and it is self-replicating. It can replicate itself. Inclusion bodies. So they are non-living structures present in the cytoplasm. They may occur freely inside the cytoplasm. Synophysian granules, glycogen granules. These are the granules that are, you can say, the nutrients for the bacterial cell, and they are stored, and whenever they are required, they can use that. Okay, or covered by the 2,4 nm thick lipid non-unit protein. Again, it is a non-unit protein membrane, and sometimes they also contain the gas vacuoles or the sulfur granules. Now, what is the importance of this gas vacuole? The gas vacuoles make the bacterial cell light. The gas is filled in it, and it makes the bacterial cell light, and they can present at the surface, right? So they can get the oxygen, they can get the nutrients, right? So this is very, very important. So these are some of the inclusion bodies that are present in the bacterial cell. Flagella, very important. Bacterial flagella are the uni-stranded equivalent to the single microtubular fiber. It is made up of 3 parts: basal body, hook. So if we talk about the bacterial cell, so here the basal body is there, right? Like this. This is the basal body. Basal body. Hook-like structure is there. Hook and a filament like this. A filament-like structure. 3 part. So this flagella is present, okay, and it is a single microtubular structure. They are made up of protein flagellin. So this is the protein that is named as flagellin and used for making the flagella. Now here comes the reproduction in bacteria. It occurs in three methods: binary fission, sporulation, and sexual reproduction. So we will see one by one what are the different types of reproduction method. The first one is the binary fission. So let us discuss. It is a common method of bacterial multiplication under favorable conditions. So under the favorable condition, what they do, the nucleus divide into two. The chromosome material divide into two. It replicate, it makes its copy. One copy is transferred to the one supposed to be a cell, and another copy is transferred to the another part of cell that is going to be a new cell, right? Now what happened, you can see the nucleoid divides, cell wall and membrane begin to form transverse septum. So here you can see a septum like structure is there, right? Septum, or you can say the invagination. This invagination goes there and it unite together, and form a transverse septum becomes complete. Now this septums here you can see it becomes complete, and the two daughter cells get separated. Now this two daughter cell is having this nucleoid structure, two nucleoid structure in their cell. So this binary fission of bacteria does not involve formation of spindle fiber. So there is no spindle fiber formation in the case of this bacterial. Now I can ask why there is a need of spindle formation for the division, and here they do not require such. Because here only one chromosome is there, and when the two chromosome is formed, two circular chromosome, one is shifted to the one pole and another is shifted to the another pole, which is very simple, right? And this invagination is also helping, so it give a pressure here to movement of this one nucleoid structure here and one nucleoid structure to the another side. But if you see in the case of eukaryotic cell, whole chromatin structure, it's divided into small fragments that is called as chromosomes, right? And each chromosome divides into two. It makes a copy into two like this, right? And each copy has to move. Now one copy has to be in this side and one copy goes to this side. So here there are so many chromosomes are there, so they require formation of the spindle formation to divide the bacterial cell having equal number of chromosome that moves to the two different cells. But here only one chromosome that replicate and makes its copy. So one copy moves to this side, one copy moves to this side. It's very simple. So they do not require any spindle fiber formation here. Okay. So this is about the binary fission. Two same type of cells has been formed here, right? Daughter cells. Reproduction in bacteria, sporulation. It undergoes spore formation under unfavorable condition. Means this spore formation occur under the unfavorable condition, means the condition that is not favorable. Harsh condition, we can say. Harsh condition. Water is not available. Food is not available. That is called as the unfavorable condition. So what does the bacteria do? Bacteria produce several types of spores called gonidia, endospores and cysts. So here you can see the primordial monoderms, okay, sporulation. It's nucleoid divide, and it's form a covering over it. Okay, retention of the outer spore membrane. This membrane is formed against this spore formation, right? And sporulating diderm. Now you can see the retention of both, loss of sporulation, loss of outer membrane, and loss of both. So any of this thing could happen. So at that time, what happened? Sometime in the harsh condition, this dies off. But what is left? This spore is left. It can withstand the high temperature, high harsh condition, right? On non-availability of food. And when the condition become favorable, this spore give out its covering and the bacteria is there. So this is called as the sporulation. This is also asexual method. Coming to the reproduction in bacteria, that is the sexual mode of reproduction. Typical sexual reproduction is absent in bacteria because of the absence of meiosis and gamete formation. So again here, no meiosis is there, no gamete formation is there. So sexual reproduction is quite difficult. Instead, gene recombination occur parasexually by 3 methods. Means there is a mixing of DNA, and that is why it is called as parasexually, and it occur by 3 different methods. Gene recombination word comes here. Gene recombination means 2 different types of DNA recombine, and that is why it is called as gene recombination. The 3 methods are there: transformation, transduction, and conjugation. So we will see one by one what are the 3 different types. It is absorption. So first one, that is the transformation. It is absorption of DNA segment from the surrounding medium by the living bacterium. So this is again a plasmid. This is a bacterial cell with chromosomal DNA. Now what happened? A plasmid or any bacteria that is comes from the another bacteria, it get enter inside it, bacteria. Okay, and the cells that have the ability to uptake DNA are known as competent cells. So this cell is called as competent cell because it is having the ability to take foreign DNA. Ability to take foreign DNA. Foreign DNA means the DNA that comes from the outer side. That is called as foreign DNA, right? So this bacterial cell is called as transformed cell, change cell. It is not the same bacterial cell. Now it got changed, and that is why it is called as transformed cell, right? So this is called as transformation. Again, any foreign DNA that comes inside the bacteria and it becomes a part of that bacteria. Now we can say this is the gene recombination. The cell that have the ability to uptake DNA are known as competent cells. So this bacteria is having the ability. It is not very always that all bacteria can take any foreign DNA coming to itself. No, some DNA they can take up. So that bacterial cell is called as the competent cell, right? Transduction. Now it is transfer of the foreign gene by the means of viruses. Now here you can see the bacteriophage virus got attached to the surface, okay, and it is giving out its DNA inside the bacterial cell. Okay, so viral DNA, and this is the bacterial DNA in the this is the bacterial cell. You can say this is the bacterial cell, and this viral, this is bacteriophage virus, it's giving out DNA inside the bacterial cell. That is called as the bacterial DNA. Now you can see a lot of fragment at the time of division. This lysis is there. Bacterial DNA get fragmented, and this viral DNA, it also makes the copies of it. Bacterial DNA fragment and viral DNA makes a copy of it, replicate. And how does it replicate? It takes the machinery, it takes the enzymes of the bacterial DNA and multiply itself, because it do not have any own those enzymes that can replicate the viral DNA. It takes up the machinery or the enzymes of the bacterial DNA and replicate it, multiply it. So now here you can see the viral DNA copies. Okay, now what happened? Sometime the bacterial DNA fragment get incorporated into the new bacteriophage. Now here you can see the red color. This one, it is having this viral genome, it is having bacterial genome. Now another bacteria, this bacteriophage virus that is containing the bacterial DNA get infect to another bacteria and incorporate its DNA inside it. Now this DNA is of whom? Virus or bacteria? It is of bacteria, right? So here you can see this DNA fragment of the bacteriophage virus that contain the bacterial DNA get incorporated into the genome of the bacteria and it become the part of this. Now here again, this is the type of recombination, genetic recombination, right? So a virus may pick up the gene of the host in place of its own gene during the multiplication in the host, and it passes over the gene of the previous host to the new host. This is called as transduction. Okay. Next is the conjugation. Now, conjugation is the method of transfer of genetic material from one bacteria to the another placed in contact. Conjugation occur in 4 steps. The number 1 step, recognizing attaching of the donor to the recipient cell. So there will be one donor cell and one recipient cell. Donor cell and recipient cell. Recipient cell. 2 types of bacterial cell. Producing the channel through which the DNA is transferred. So a channel is formed here, and this channel is formed by sex pili. Sex pili. We have discussed about this, right? And generating the single-stranded DNA that is transferred. Now from here, the single-stranded DNA is transferred to the another bacteria. Establishment in the new host, and in that it get established in the new host. So here you can see this is the F plasmid, this is the recipient, and this is the donor cell. Okay, now the sex pili is formed here. The sex pili is formed, get attached, right? Now this F plasmid, it's replicate itself, okay, and makes its copy, and it is transferred to the recipient cell that do not have any plasmid like structure. Okay, so this it happened in the form of conjugation. So again, this is a type of sexual reproduction. It is not actual sexual reproduction. Here it is called as sexual reproduction because transfer of a DNA that is from one cell to the another cell is occurring here. There is no gamete formation is there. There is no meiosis process is taking place, right? So this is not an actual reproduction, sexual reproduction. The transformation, transduction and conjugation are a type, we can say, right? So this is a very, very important. Now, some of the bacterial disease that occur in the crop plants. So here we are having a table that is having a disease, causative agent, host and the symptoms. So fire blight, first disease that is called as fire blight, and it is caused by the bacteria that is called as Erwinia amylophora, and the host are apple and pear. What are the symptoms? Blossoms are water soaked. So all the blossoms, that is the flower, that become water soaked, and that can do not give a new you can say fruit, that do not forms the fruit. So Erwinia amylovora, that is called as fire blight. Crown gall, that is caused by the Agrobacterium tumefaciens. This is a bacteria, right? And more than 100 genera they are found, in more than 100 genera, including the herbs, shrubs, and even the hard wood tree. Small enlargement of stems or roots can be seen basically at the level of the soil. So this is very important, right? Enlargement of the stem, and that forms a crown or a gall-like structure, and that has got its name, crown gall. So this is the second type of disease. Next is the wildfire of tobacco, that is caused by the Pseudomonas syringae. Pseudomonas syringae, that is a host that is called as tobacco, and yellowish green spots on the leaves that can be seen in this case. Okay, wildfire of tobacco, Pseudomonas syringae. And the next one is this is the third one, and the fourth one, that is the citrus stubborn disease, that is caused by the Spiroplasma citri. Spiroplasma citri. This is another bacteria, and it hosts our citrus and some stone fruits. Chlorosis, yellowing of leaves. So generally the leaves become yellow and then do not perform the process of photosynthesis, and the food consumption is also become reduced. Fruits are not good in quality. So all these happen in this disease. Okay, so this is all about the bacteria, that is the one of the microbes. Okay, so we have completed this session. We will have some more session to complete the microbiology in agriculture. Till then, thank you very much.